Good afternoon, everybody. I'm Al Sommer and have the pleasure of chairing the Vision Prize jury for the Chamapalimaud Foundation. I would like to thank ARVO's trustees and leadership for making these ARVO Chamapalimaud lectures a regular and distinguished part of the ARVO annual meeting. The Chamapalimaud Vision Award recognizes fundamental discoveries in vision research and on alternative years, innovative service programs for the prevention of blindness in the developing world. Both awards carry a stipend of €1 million, which is meant to assist recipients in furthering the work for which they are being recognized. The 2012 award, which we celebrate today, was bestowed upon the recipients at a special ceremony in Lisbon that was presided over by the President of Portugal. This year, the jury received over 30 outstanding nominations. If you nominated a group that did not win the prize this year, please resubmit an updated nomination in future years. Given the competition, this just may not have been the year for your nomination. This, the sixth year of the award, recognizes important scientific breakthroughs that have improved our understanding of how the eye functions, how disease leads to blindness, and how eye disease can be more readily detected and treated so as to protect and preserve vision. Vision research commonly honors discoveries that improve the ability of the eye to visualize the world. Such was the case when the first Chamapalimaud Vision Research Award recognized the contributions of Jeremy Nathans and Qingwei Yao for their discovery of the way light rays are detected by retinal cells and converted into neural signals sent to the brain. And the second Chamapalimaud Vision Research Award recognized the work of Anthony Movshan and William Newsom for their elucidating the way in which the brain's cellular networks convert these light-induced retinal signals into discernible patterns that we call vision. This year, the third Chamapalimaud Vision Research Award breaks with that tradition. Instead of recognizing discoveries of how the eye visualizes the world, we recognize instead discoveries that have improved our ability to visualize the eye. The first and perhaps last great advance in instruments for visualizing the structures inside the eye was Helmholtz's ophthalmoscope, introduced in 1851. It revolutionized eye care by enabling us to see and recognize abnormalities of the eye and its structures that caused disease and led to vision loss. While there have been many improvements and modifications of this essential instrument since then, there have been very few radical new approaches to visualizing the inner eye. Until, that is, the work of this year's Chamapalimaud laureates. David Williams has spent his career adopting the technology of adaptive optics to eye research. Adaptive optics were originally developed by astronomers to help them better visualize the heavens by correcting for the many distortions induced by the Earth's atmosphere. David Williams pioneered the adoption of adaptive optics to visualizing the retinal photoreceptors in the back of the eye. His techniques neutralize the many complex aberrations that distort light rays transiting the ocular structures on their way to illuminating the retina and on their way back to the equipment recording the retina's appearance. Williams' techniques have so dramatically increased the clarity of the retinal images that individual photoreceptors can be visualized and followed over time in living human subjects. By documenting changes in the appearance of photoreceptors over long periods of time, his techniques have dramatically advanced our understanding of retinal changes in aging and disease. Our second and related 2012 Chamapalimaud laureates developed a revolutionary way to record the appearance of the ocular structures of patients in cross-section, beginning with the retina, now including the optic nerve and the anterior segment of the eye. The fundamental technique, optical coherence tomography, was developed and applied to studies of the eye by the productive team of James Fujimoto, David Wong, Eric Swanson, Carmen Pugliafito, and Joel Schumann. Within an astonishingly short period of time, OCT has become a basic tool for detecting and managing a host of ocular conditions and a core piece of equipment in any modern ophthalmic clinical facility. Both of these new techniques have revolutionized ophthalmic practice and our understanding and management of ocular changes in aging and disease. Please join me in congratulating Dr. Williams for his innovative application of adaptive optics and Drs. Fujimoto, Puliafito, Swanson, Wong, and Schumann for the development and application of OCT on their richly deserved receipt of this year's Antonio Chamapalimaud Vision Prize. Stand up, guys. As you all know, David will be delivering the Friedenhall Lecture tomorrow afternoon. Dr. Williams will discuss imaging single cells. I'm sorry, you can sit down now. David will discuss imaging single cells in a living eye, after which Dr. Fujimoto will speak on behalf of the OCT team, titled Optical Coherence Tomography, Translating Technology from Research into Clinical Practice. David?
Thank you very much, Al, for that generous introduction. Lenore, thank you so much and your foundation for this unbelievable opportunity. It's a really amazing experience to have the chance to speak to you today. And I wanted to emphasize that the committee gave this award to the Williams Laboratory, not to me. You'll see as we go through this presentation this afternoon that there were a large number of people from many different laboratories around the world who participated in a community, in a very collaborative effort to generate the results that I'll be showing you today. And I just wanted to acknowledge all their contributions to the work that I'll be telling you about today. Now, our agenda for the last couple of decades has been to increase the resolution of the ophthalmoscope. And to give you a sense of the spatial scale that I'm talking about, I have to disclose my commercial relationships first. And then consider an eye such as this one. If we were to put a 10 millimeter square on this eye, just for a sense of scale here, and then follow that into the eye, that same size square would look something like this, which is roughly corresponding to what a conventional fundus image is. But if we look 2 orders of magnitude finer, that's the spatial scale where adaptive optics instruments operate. Now, looking at this conventional fundus image in that way, you can see there's precious little spatial information in this image. It's almost devoid of any spatial detail, and that's for a lot of reasons. And over the past 15 to 20 years, we've been developing a lot of different ways to enhance the information that's available at this spatial scale, where we're now looking at 100 microns instead of 10 millimeters of tissue. And one of those technologies is adaptive optics, but there are actually many involved, and I'll as we go along, you'll see, you'll get glimmers of some of the other aspects of this that have allowed us to achieve the resolution that we can. But if we were to look at the same patch of tissue with adaptive optics, this is the kind of image we would see. Here, this is taken in a living human eye. All of our work is designed to enhance the ability to view the retina of a living eye, so that we can do studies that were otherwise not possible through excising the retina and looking at a microscopic spatial scale that way. So here you can see an image that shows you the rods and cones. The rods in this case are the small cells, and they're about 2 microns in diameter. They're quite close to the current limits of transverse resolution of our instrument. And as I said, this is due to a number of features, including using monochromatic light and correcting for eye motion. You're seeing an image that is really the average of many images that have been registered to take out the effects of the blurring effects of eye movements, and also one of the key technologies has been adaptive optics. So I want to take a minute to explain to those of you who don't know how that works, how it does work. We borrowed this technology from astronomy. You're looking at the Keck telescope here, which is a telescope that has equipped adaptive optics and achieves high-resolution images exceeding those of the Hubble Space Telescope in some cases on the ground by virtue of having adaptive optics in it. Our first attempt to translate this technology into the eye was in 1997 with Don Miller and Zhizhong Liang in my laboratory at Rochester, and we published a paper then in which we showed a couple of benefits of adaptive optics for the eye that I'll go through for you. But first, let's take a look at what the fundamental problem is. If we all had perfect eyes, this is what would happen. If we put a small beam of light into the eye like this, it would hit the retina, be focused on the retina, hit the retina, and come back out, and we end up with a really beautiful wavefront emerging from the eye that was nice and planar, and that's just what you want for high-resolution retinal imaging. But in all, realize there are higher-order aberrations. In addition to the aberrations that glasses can correct, and the situation that actually emerges is this. The light passes into the eye and comes back out, but as it passes through the lens and cornea on the way out, there's a distortion in that wavefront that is what we call the wave aberration. In this cartoon, I've exaggerated this by about a factor of 1,000. It's typically really a deformation in that planar wavefront that's on the order of a micron or less in a real human eye. But to illustrate it for you, I've exaggerated it considerably. Okay, well, so what do we need to do to fix this problem? Well, we need two things. We need to be able to measure the wave aberration, and we need to be able to correct it. So let's look at how we measure it first. The device we use to measure the wave aberration, there are various variants of this. I'm going to tell you about the one we have been using in our laboratory, the Shack-Hartman wavefront sensor, which consists of two components, a lenslet array in the front of the In the front here, I've just shown you a small number of lenslets. Typically, there are several hundred of these in a real wavefront sensor. And then there's a light sensor, a CCD array back here, that collects a bunch of images of the retina. When we cast a wavefront onto that, emerging from the eye onto the lenslet array. The lenslet array breaks that wavefront up into a number of sub-wavefronts, each of which forms an image on the CCD array. And when you have a perfect planar wave hitting the wavefront sensor, you get a beautiful regular array of spots on your CCD. And that's how you know you've got perfect optics, which unfortunately none of us have. With an aberrated wavefront, however, the wavefront hits the lenslet array, and the slope of the wavefront varies from lenslet to lenslet, so the spots become irregularly arranged on the CCD detector. We can measure those displacements, and from that we can reconstruct what the wave aberration is. Well, how do we correct the wave aberration? We now know what the shape is of that deformation, which is different in every eye. How do we correct it? We use a deformable mirror for that purpose. You're looking at a deformable mirror head on here, and you can see that that mirror is warped. And the warping that we have implemented in this particular case is one that captures exactly the shape of the wave aberration, but at half the amplitude. And that's just what you need so that when a wavefront strikes the deformable mirror, it will reflect off of it as a plane wave and correct it. We use that light then that's now passed, some of which has passed through the wavefront sensor and the majority of which has gone through the deformable mirror and into a camera in order to take pictures of the retina. So just to see the whole system now put together, here's the eyeball we're imaging, wavefront sensor over here, deformable mirror for correction over there. Away we go. We deliver a beam of light into the eye, comes back out, reflects off the deformable mirror where the mirror's not set right, so it's aberrated. Wavefront sensor makes a measurement, corrects the deformable mirror, and we repeat this process multiple times. In fact, while we're doing these measurements, the wavefront sensor and deformable mirror are running in closed-loop mode at 30 times a second roughly, or something like that, all the time throughout the measurement. Then we put in an imaging flash like this. That imaging flash then is directed to a light sensor at the back, and that's where we acquire our high-resolution retinal image. Now, I want to go back, though, a few years to the very first images that Don Miller took as a graduate student in my laboratory, images of cones in a living eye without adaptive optics. So it turns out you don't need adaptive optics to see cones, as Don showed before adaptive optics existed for the eye, just about a year before it existed. He published this paper. And just to fast forward then to 2011, here's an image of the very center of the fovea obtained by Alf Dubra with a state-of-the-art current adaptive optics instrument. You can see the very smallest cones at the very center of the fovea, and compare that at the same spatial scale with Don Miller's image on the left. You can see how far we've come. What's been remarkable to me is how incremental, in a way, this process has been. Everyone who comes through our laboratory and other laboratories around the world have been making small tweaks to these instruments that over time accumulate and snowball into great substantial improvements in not only the quality of the images, but the robustness with which these instruments operate. And that's very important for clinical application, which, of course, is one of our most important goals. Now, I wanted to explain one other feature of these systems, which is pretty nice. And that is that, suppose we take this light sensor, this camera in the system, and we replace it with a light source, and we project light back in the opposite direction through the mirror. You might think, oh, no good, we've got now entering the eye a deformed wavefront. That's not right. But it turns out if you understand the principle of the reversibility of optics, that's just the right wavefront to give you a really sharp focus spot on the retina. So we can actually run adaptive optic systems in reverse, and we can deliver light stimuli to the retina that have unprecedented resolution and sharpness. And that allows a whole range of experiments on vision and vision correction that haven't been possible before. When we first demonstrated this in 1997, there was a lot of excitement about it because it allowed people invested in laser refractive surgery to get some sense for the possible benefits they might be able to achieve if they could do refractive surgery at high and correct higher order aberrations. And so instead of using a deformable mirror, their strategy, of course, was to measure the wave aberration with a wavefront sensor and then correct it with an excimer laser rather than a deformable mirror so that you have a permanent correction in the eye. I would say that in the history of refractive surgery and the use of wavefront guided techniques, it's been very helpful, but probably the most helpful thing is just to provide a very clear description of what's not quite right or what's not quite optimal in laser refractive surgical techniques so that they can be improved. And so while wavefront guided surgery hasn't provided us all with supervision way better than we could possibly imagine, it certainly has improved that technology enormously over the years. Another interesting application for vision correction of wavefront technology is in customized contact lenses. Gunyang Yoon at the University of Rochester has been invested in this for some time. And here's an example of a soft contact lens for a keratoconic eye that can correct the wave aberration, reduce it by a factor of 3, and give 2.1 lines of improvement in visual acuity in a keratoconic eye, which is a substantial improvement and well worth trying to achieve ultimately in a commercial product. So Gunyang can do even better than this with scleral contact lenses and has even better control over the wave aberration for vision correction. Now, another application, going back now not to the use of other correction devices like contact lenses or intraocular lenses, which are also used here, or refractive surgery, but going back to that adaptive optics instrument, there's been a lot of activity to use adaptive optic systems as vision stimulators. So not only can you correct the aberrations and do studies psychophysically of how well the nervous system can see when the optics of the eye are completely corrected, you can also use this device to generate aberrations. So you can study the impact of aberrations of any kind on visual performance in psychophysical testing. And one of the pioneers in this has been Pablo Artal, who incidentally was also very instrumental in helping us develop the first automated wavefront sensor for the eye, and has a long history of work in this area. You can see Pablo in the upper left there. Here's Pablo. And an example of just one application of this is he and I collaborated on a project and others in which we rotated the aberration of the eye. So someone would look at a subject in this experiment, look at the world through either their normal wave aberration or a rotated version, which had exactly the same amount of aberration, but it was rotated to the wrong orientation. We found that the world is far clearer when people look through their own native wave aberration in its proper orientation, indicating that there is substantial neural adaptation to higher order aberrations in the eye. The brain accommodates to these higher order aberrations, and there's a very active program. A number of investigators around the world are looking into that further and exploring their implications for the implications of that adaptation process for refracting the eye. Okay, now finally, it gets even better than that for visual psychophysics, and the reason is because, and this really caught me by complete surprise, but Austin Rorda and Scott Stevenson and others realized that when you collect a retinal image with adaptive optics, you actually have an exquisite measure of how the eye is moving. And on the left here, you can see a letter E projected on the retina, and you can see the real motion of the eye at a cellular spatial scale during normal fixation. And what they realized is that they could take that information and turn it around and develop an eye tracker far superior than what has ever been developed for tracking the eye before. And an example of that is shown here, where on the right you can see a stabilized video, and so that E is stabilized, and those of you who are perfectionists in the audience may say, well, it's still jittering a little bit. And you're right, but it's getting better all the time. And that jitter, after all, is on the spatial scale of less than a cone. So we're now able, and Austin and his group at Berkeley are able to deliver visual stimuli to the retina and control exactly what photoreceptors get stimulated when they do that. And that's useful not only for psychophysics, but also for visual physiology. So I think we're in an era where we'll see a whole new generation of much better controlled psychophysical experiments, and also clinical experiments. This is sort of the ultimate microperimeter, right, where you can actually deliver a stimulus to a single photoreceptor and ask whether that photoreceptor is viable or not. And so very small defects early in the development progression of a disease are trackable potentially with technology like this. Well, now I want to return to the retinal imaging applications of adaptive optics and give you an overview of some of the more exciting applications. One of the first things that Austin Rorda and I did at Rochester, and then Heidi Hofer, who joined our team later, also spent quite a lot of energy on, was trying to understand what the geometry, the packing arrangement is of the three kinds of cones in the eye. This had been a controversy for 200 years since Thomas Young first proposed that there were three kinds of photoreceptors in the eye. And finally, with adaptive optics, we were able to image the cone mosaic. By combining it with retinal densitometry, we could identify the photopigment in every cone photoreceptor, and that allowed us to see for the first time the geometry of all three cone classes in the eye. What was even more gratifying for us about this is that we could do it in living eyes, non-invasively. So we were able to do color vision experiments I don't have time to explain in tandem with these imaging measurements and study the behavior of different mosaics with different properties. Now, shortly after that, Joe Carroll joined the lab as a postdoc and then rather quickly moved on to an independent career that many of you, I'm sure, have been following. And Joe discovered Joe's whole strategy, and this was really based on his graduate work with Maureen and Jay Knights at the Medical College of Wisconsin then. And the whole game there was to measure the genotype and to do phenotypic comparisons with the genotype. And so here's just one example in which you can see the mosaic of a normal trichromat on the left. And if you were to look at the mosaic, for example, of someone who is a single gene dichromat, it would look the same. That is, a single gene dichromat has only two pigments instead of three, because they only have two genes instead of three, but they have a normal appearing mosaic. But there are cases of color blindness, such as you can see on the right, a deuteranope who has a mutation in the M pigment gene, and there you can see, I hope, gaps in the mosaic that represent the loss of at least healthy photoreceptors, and we've done psychophysical experiments that confirm that there are little microscotomas where those cones are dark in that image. So you can see, I think, the potential of this in clinical work if we can track the loss of photoreceptors in a progressive disease, which fortunately this is not. But if we could do that, we have the potential of catching, by having the ability to view the retina at a microscopic spatial scale, at cellular resolution, we have the capability of seeing very early changes caused by retinal disease before it's so severe that treatments won't ever be, may never be a possibility. Now, just to sort of amplify on that point, I wanted to call your attention to an interesting study that came out not too long ago that is a collaboration between Jackie Duncan's group at UCSF and Austin Rorda's group at UC Berkeley, where they have been studying the efficacy of ciliary neurotrophic factor treatment, CNTF, in the eye, and have shown by counting cone photoreceptors in RP patients and going back to the same patients month after month after month, they can see that this particular treatment has been effective in rescuing photoreceptors from loss, whereas the fellow eye, which was untreated in this case, and I won't show you all the data here, continued to show the progressive onslaught of the disease. So I think there's going to be an interesting future for measuring counting cones in patients with a variety of different kinds of diseases, including macular degeneration, in order to understand what the progression of that disease is, and also to see whether to accelerate the development of drugs to treat that progression. Now, one of the really additional things about adaptive optics imaging that's gratifying for us is that you can combine adaptive optics with whatever your favorite other imaging modality is. You can use adaptive optics with it pretty much. And here's an example from Steve Burns' work in his laboratory at Indiana University. Where on the left you can see he's combined dark field imaging with adaptive optics imaging, and you can see on the left flow in a relatively large vessel by our standards here. You can even see the heartbeat if you look carefully at the change in the velocity in that vessel, and you can also see what are parasites that ensheath that single vessel. On the right, you can actually see single red blood cells flowing through single capillaries with this method. We think this is going to be very exciting in diabetic retinopathy, and groups are already heavily invested in using this to study the changes in the vasculature at the very finest spatial scale that are caused by diseases like diabetic retinopathy. Now, here's another example of a technology that can be combined with adaptive optics with interesting outcomes. And this is from the work of Don Miller in his laboratory at Indiana University. So let me take you through this. So what Don has done is combined adaptive optics with OCT. You'll be hearing a lot about OCT shortly, of course, from Jim. And so in this image you can see, I'm blowing up, or Don is blowing up this image here, and you can see that if you plot this linearly, not using the logarithmic scale that's usually used in OCT images, you see that there's really a lot of the light coming from just two separate layers in the photoreceptor. Each pair of reflections here comes from a single cone photoreceptor in a living human eye, and the upper one, the upper reflection, here's one and here's the other. The upper reflection comes from the inner segment, outer segment junction of the photoreceptor. The lower reflection comes from the posterior tip of the outer segment. And because you have two reflections coming back from one structure, if you put coherent light onto the retina, those two reflections will interfere, either constructively or destructively. So that's neat, because what that means is that if you track the retina over time, you can see scintillations in the light output of individual cone photoreceptors. I think this is tracked over a 5-hour period, a heroic experiment for one of Don's graduate students, presumably. And we stopped doing that. Our human subjects people won't let us do that sort of thing in our lab, but anymore. But this is exciting because these variations in reflectance actually tell you about something important that's going on in single photoreceptors in living human eyes. And what that is that if you track this sinusoidal oscillation in these cones over time, you can compute that there's a change in the outer segment length that is moving along quite smoothly here with a slope equal to 123 nanometers, not microns, not millimeters, nanometers per hour. And so what that means is that Don is actually able to measure changes in the outer segment length as new discs are added to the outer segment and you can see a change as small as 45 nanometers, which is just slightly thicker than a single outer segment disc. So Don is approaching the level where he could see the addition of a single disc to the outer segment of a single cone in a living human eye. And I think that's a remarkable thing. That number of 45 microns is nearly it's 600 times less than the current resolution of the best OCT system. So it's a very specialized situation, but it's an example of the kind of precision that can be achieved in combining adaptive optics with other imaging modalities, in this case the delivery of coherent light to the retina. Now, in our group, we've been very heavily focused on fluorescence imaging. Fluorescence imaging is something you can't do with OCT, and we have been hoping to capitalize on the revolution that we're in the middle of. And unfortunately, we were going to hear about that tonight from Roger Chen. He was unable to be here. But there's really, as many of you know, a remarkable transformation going on in our ability to develop fluorophores that target specific cell classes and tell us interesting functional things about those cells in the brain or the retina. And I'd like to take you through some of the work that we've done that aspires to, as I say, capitalize on that revolution in the development of various molecules for reporting the activity of neurons. One of the first things we did was to rely not on extrinsic fluorophores, like the ones I was just talking about, but to rely on lipofuscin, which is endogenous to the retina and resides in the RPE. And Jessica Morgan was able to take images in monkey, anesthetized monkey here, of the RPE mosaic. And if I blow this up, you can get a little bit better feel for what she can see there. Here you're seeing a single RPE cell. This is the nucleus right there, and it's dark because there's there's no lipofuscin in the nucleus, but there are granules of lipofuscin in the cytoplasm around the nucleus. So you see these hot spots that correspond either to large granules or clusters of granules around the edge of the cell. So that allows us to see lipofuscin, and we're and RPE cells, and we're excited about using this, and Ethan Rossi at Rochester is using this now to look at macular degeneration and watch the progression of changes in RPE in that very devastating disease. Also, Melissa Gang showed that you can image using now not endogenous fluorophores, but using GFP, and which we've gotten into cells using a viral vector, an AAV vector, and expression in cells. Here are some examples of some of her images in the mouse. So in addition to imaging primate, we can image mouse retina. Mouse turns out to be much better than any of us, certainly than I previously thought, as an imaging model, because it has twice the numerical aperture of the human eye, and it also that means it has twice the resolution and 4 times the light collection efficiency. So we can see individual ganglion cells, and we can begin to do classification of ganglion cells even in living eyes. Now, one of the questions that we're very interested in addressing someday in our group is, why do we have so many separate channels to convey the retinal image to the brain? And many people in this room are interested in that idea and exercised about how to solve that problem. One of the reasons we've made a lot of progress on that, but one of the reasons we haven't made more is that we haven't had the ability to record from large numbers of ganglion cells in the living eye. So we've been working toward that with Lou Yin and Bill Merrigan at Rochester. Here you can see Lou has encircled 80 ganglion cells in the monkey fovea, corresponding to the very center of the fovea. And we can record from all of those cells simultaneously by putting in not GFP, but a reporter molecule that is a calcium indicator, one of the variants of GCAMP. And you can see here optical responses to individual ganglion cells generated by imaging many soma simultaneously. I'm just showing you 6, but we can do 80 now simultaneously. There's fundamentally no reason why we shouldn't be able to do thousands of these ultimately, simultaneously, which will allow us to look much more carefully for these very rare cell types that are difficult to find with a single microelectrode. And even a microelectrode array, while it can record from many cells, doesn't have the capability of allowing you to do this in a living animal over many, many months. So if you're interested in, or even years perhaps, if you're interested in development of the retina, or if you're interested in changes over time in some disease of the retina, it's conceivable this method may prove very valuable for studying that sort of thing. Now, I wanted to give you a couple of other examples of how limits in imaging the eye can be broken. One limit that you might wonder about is the spectral limit. And if you look at the white curve in this figure, that's the ocular transmittance. That shows you the range of wavelengths that you can get into the eye. But if you were to try to stimulate the retina with ultraviolet light in an intact living eye, like we like to do ultimately in humans, you run into a problem because there's essentially no transmittance in the UV. So you might think that molecules like NADH are completely off limits, inaccessible in conventional imaging. But in fact, that's not true. We can use another method that's been around for quite some time, 2-photon imaging, but hasn't been applied in this way and has never been applied in the primate eye before. And we can deliver infrared light into the eye, and because of the nature of the nonlinear nature of 2-photon imaging, 2 photons of infrared light behave as 1 photon in the UV, and you can excite that fluorophore which is an interesting metabolic marker. And the fluorescence is at longer wavelengths, so it comes back out through the window. So you can put light in through the window, you can get light out through the window, but you're interrogating molecules that are otherwise inaccessible with conventional imaging methods. And Jennifer Hunter at Rochester has demonstrated that this method is viable, even though our efficiency isn't very good yet, but it's viable in the primate eye. And these are the inner segments of an array of cone photoreceptors in a living monkey eye obtained with 2-photon imaging. We're excited about this because every cell class in the retina has a strong, well, it's a weak signal, but it's a high contrast signal in 2-photon imaging. And it may someday be possible in human without the use of exogenous fluorophores to see these cells if we can make this an efficient technique, which currently is not, but we have hopes that that will be possible someday, and it's certainly a challenge to work toward. Now, to conclude, I wanted to remind everybody that for many, many years, Ernst Abbe had proposed the diffraction limit in microscopy. We've corrected the aberrations with adaptive optics. Confocal imaging that Austin Rorta added to adaptive optics takes care of light scatter. But there's one major source of image blur that we're not addressing, and that's diffraction. And I taught students for years and years that that was a fundamental limit having to do with the wave nature of light and something we would never, ever surpass. Forget it. Go home. Think about some other project. And, of course, I was completely wrong. And fortunately, we have a place where we can put assumptions that are cherished and near and dear to us, but we can lay them to rest. And we should do that in this case because, in fact, there are 3, at least, techniques that are viable candidates for using the human eye that haven't been, to my knowledge, used successfully, although I know there's work reported at this meeting in this area. Structured illumination, stimulated emission depletion, and fluorescence photoactivation localization are all ways, bona fide ways of surpassing the diffraction limit. So I think it's just a matter of time before we could see other things like dendritic spines and really drill down on the circuitry in the retina to understand how it works using these fluorescence methods and so on in combination with this. So I think there's a very high hope for the future that adaptive optics isn't the end at all. It's just one small tool in a whole range of tools at our disposal to look at the retina in novel and exciting ways. Thank you very much.
I neglected to do something. Could you turn on the hall lights for a moment? Is that possible? Yes, thank you very much. There are a number of my fellow jurors who are here, and I neglected to ask them to please stand and be recognized. So, Paul, Nag, Jose, can please. Thank you. Okay, James, I think you're up next.
Well, it's a great pleasure to have the opportunity to represent our collaborative team of David Wong, Carmen Puliafito, Joel Schumann, and Eric Swanson. In this presentation, we'll be describing technology from a different viewpoint, translating from research to clinical practice. Here are our disclosures. And before I begin, we wanted to especially express our thanks and gratitude to the Chamapalimaud Foundation, as well as the president of the foundation, Leonor Beleza, and the award selection committee. It's really a great honor to be selected for the Vision Award. I think the Chamapalimaud Foundation is an inspiration to all of us in the research as well as the clinical community because it symbolizes or embodies the translation of research to clinical care, and the Vision Award itself describes both fundamental research as well as the delivery of clinical care on an international basis. I also wanted to mention that Dr. Wong will be presenting the Friedenwald Award lecture that's scheduled for tomorrow at 6:30. So optical coherence tomography is a method of imaging which approaches resolutions that we can assess with histopathology, but allows imaging in real time non-invasively. And perhaps the most developed application would be ophthalmology, but there are also applications in intravascular imaging as well as in fundamental studies such as developmental biology and genetics and endoscopy. So it's a broad-based imaging technology. We actually started working on biomedical optics from the viewpoint of femtosecond lasers or ultra-fast phenomena. This shows an example of femtosecond laser technology at MIT dating back to 1983. And at that time, you can see that these are quite large systems. These were so-called dye lasers. This is a femtosecond ring laser and an amplifier. And Carmen Pugliafito and I were working together using this technology to assess retinal injury. But perhaps the first suggestion for using light to image in biological tissues was work by Michel Duguay, who was at AT&T Bell Laboratories. This is a paper dating back to 1971. And in this paper, Duguay shows an ultra-high-speed shutter that's actually a device here that is a laser-activated shutter with cross-polarizers. And he uses this to photograph a pulse of light that's propagating through a glass of milk. Actually, here you see the light pulse. So it's an example of a very high-speed photography operating close to a limit. And in the same publication, Duguay commented on the ability to see behind scattering material. So he had an AT&T logo, and then he placed it behind a scattering piece of paper. And then by using the high-speed shutter, he could gate out the unwanted scattered light and partially recover the image from in back of the scatterer. So in his paper, he commented that one interesting possibility of so-called gated picture ranging is the potential to see through the human skin. So the concept that you can actually start to do histology, let's say, non-invasively. So we were very interested in this, and we applied our femtosecond laser technology using so-called cross-correlator. This is an instrument that takes the laser pulse. It measures the reflected or backscattered light, comparing this to a reference arm using a nonlinear crystal. So with this, we can have very high resolution on the order of 65 femtoseconds or 15 microns. And here we see a measurement, actually this is an ex vivo measurement of a bovine or cow eye, where we see reflections from the anterior surface, that is the air cornea interface, and then the cornea aqueous interface. In this case, the sensitivity was 70 dB, and actually that's not enough really to see the retina. So the next advance was using low coherence interferometry, which was in fact well developed in telecommunications. In this case, we avoid the use of a femtosecond laser. We have a light source and a Michelson type interferometer with one path going to a sample, a second path going to a reference. And then by scanning the interferometer and using either low coherence light or a short pulse, we can localize a reflection to extremely high time resolution, essentially femtosecond resolution, but using low coherence light. The advantage of this method is that it has extremely high detectivity on the order of 100 dB sensitivity, which is, by the way, typical for OCT systems used clinically. And with this, we could measure very weak reflections from actually inside the cornea as well as from the lens. And then by translating the beam, we could measure a different dimension to the iris of the eye, for example. Well, the first demonstration of OCT dates back to 1991, and this was proposed and developed by David Wong when he was an MD PhD student in our group. The concept was to scan the optical beam in the transverse direction, similar to ultrasound, and display the image as a false color scale, which you see here. This is an ex vivo retina. You can see subretinal fluid, and because it's ex vivo, we can have corresponding histology. So this image was taken with a 10-micron resolution. And I want to show this early photo of David Huang, who is an MD PhD student. This is really intended for the trainees and graduate students in the audience, really to show that it's possible to make significant advances early in your career. In parallel with this, there was an independent description from Japan by Professor Naohiro Tano. He described a patent measuring light wave of reflected image, which also describes the concept of image construction. So the next step was to develop technology which could be used in the clinic. And I want to especially acknowledge the contributions of Eric Swanson because he's not as well recognized in the vision community. Eric's work was critical to the clinical validation of OCT. Eric was working at MIT Lincoln Laboratories at the time. You see a photo of him here. And he designed and developed the first clinical OCT system, the electronics package, the patient user interface. And he's also author of one of the first OCT patents. Eric later went on to become co-founder of several organizations, Advanced Ophthalmic Devices, which transferred OCT technology to Carl Zeiss, as well as Sycamore Networks. This is a network company that went public, Light Labs Imaging, for the commercialization of intravascular imaging, as well as a number of other organizations. Well, using this type of technology, it became possible to perform the first in vivo retinal images. Here we see an example from our group at MIT, and in parallel and independently, the group at the University of Vienna, led by Professor Fercher, also described the first in vivo OCT imaging. Both of these results date back to 1993. So with this technology, it became possible to image both the anterior eye as well as the retina. Here you see an example of the first image of the anterior chamber. This was taken by Joseph Izat, who was in our group at the time. By the way, this is Joe's eye. Joe later went on, as you know, to become a leading eye researcher and in the ophthalmic community. And this is an example of an early retinal image, which shows the retinal contour as well as the normal optic disc profile. So using this system at the New England Eye Center in clinical studies led by Carmen Pulifido and Joel Schumann, it was possible to image over 5,000 patients and perform the first studies of glaucoma, diabetic retinopathy, and macular degeneration. And I especially wanted to highlight the work of Michael He, who was an MD PhD student at the time. He was involved in these studies and developed many of the software protocols that are actually in use in the Zeiss instruments today. So we transferred the technology to industry. We were very fortunate to work with Zeiss. They developed multiple generations of the technology, starting from the OCT-1, which you see here, and then we saw the Stratus OCT introduced in 2002. That was about the time we saw wider spread clinical acceptance of the technology. I wanted to especially acknowledge the role of industry and industrial development in OCT. This is a photograph of John Moore, who was the president of Humphrey Zeiss at the time. And I think at the time, really dating back, it was not obvious that OCT would be commercially successful. And I think Mr. Moore was really a visionary in acquiring this technology, but not only that, making the significant investment, multi-millions of dollars to really develop it. And also I wanted to acknowledge Jay Wei, who was the project manager and lead engineer for the development team at Zeiss. Jay later went on to found an independent organization, OptoView, which was the first organization to introduce Fourier domain, or so-called spectral OCT. So here we see examples of the early OCT images. This is 10-micron resolution image taken from the first generation instrument, similar to the prototype that we had at MIT. In comparison, we have ultra-high resolution OCT, and I especially wanted to highlight this because this is work that was done by Wolfgang Drexler. Ultra-high resolution, I think, was significant because it started to approach the level of histology, and it enabled us to understand and better interpret some of the clinical images at the time. So here on top we see the Stratus OCT image, 10-micron resolution, and in comparison an ultra-high resolution OCT image. In the ultra-high resolution image we can identify the major retinal layers, and we can also use this to guide the interpretation of the Stratus images. The next advance, though, came with the development of high speed. So this is the so-called spectral or Fourier domain detection methods. There's a fundamental trade-off between speed and sensitivity, but Fourier domain detection enables us to improve sensitivity and therefore to improve speed dramatically. This enables a 50 times or greater enhancement in speed compared to the earlier generations of OCT, and it's equivalent to a 20 dB increase in sensitivity. So using spectral domain detection, we can start to acquire video rate images. Here you see a repeated image near the optic nerve head. We can see the nerve fiber layer and then vasculature with blood flow. This happens to be an ultra-high resolution image at about 3 micron axial resolution. The spectral domain detection actually has quite an early history. It dates back to work at the University of Vienna in 1995. But only with the development of high-speed camera technology did it become possible to actually image the retina. The first studies were done by the group in Poland, led by Maciej Wojtkowski at Copernicus University. And then independently, a year later, several groups at the University of Vienna, the Massachusetts General Hospital, and Duke University recognized that there was a dramatic improvement in sensitivity, which would allow a corresponding improvement in speed. So at that point, many groups became involved in spectral Fourier domain OCT. Here you see an example of a high-speed, ultra-high resolution image. You can actually see pulsatile blood flow here on the central retinal vasculature, and then also we very nicely resolve the retinal layers on the level of architectural morphology. In Dave Williams' earlier talk, you see that adaptive optics can really push this to the limit of cellular resolution. But one of the big advantages of high speed was the ability to acquire comprehensive volumetric data on the retina. So by acquiring multiple scans like this, we can have a volume. The volume can be projected in different axes. For example, summing in the axial direction essentially generates an image of the retina itself. And that's extremely powerful because it enables us to generate a representation of the retina, which precisely and reproducibly registers individual OCT images to fundus features. That enables the tracking of focal changes over time for longitudinal studies. Having volumetric data also enables us to extract various data sets such as the circumpapillary scan, which is used for the assessment of glaucoma. And the advantage here is that one can compensate for eye motion in post-processing. This is a virtual scan extracted from a volumetric data set and then measured to characterize the nerve fiber layer. So by using these approaches, one can reduce eye motion and essentially improve reproducibility. It's also possible to perform longitudinal measurements, as we mentioned. This is an example from Zeiss in work pioneered at Bascom Palmer, Giovanni Gregori, Phil Rosenfeld, and Carmen Puliafito. This shows examples of mapping the retinal structures, retinal thickness, ILM, and RPE, progressing over time. So using these types of methods, one can track changes in focal pathology for a more sensitive assessment of either disease progression or pharmaceutical response. It's also possible to display volumetric data sets in an en face view. I think this is becoming more and more powerful clinically as imaging speeds increase. We have nice correspondence with fundus features. We can extract different retinal layers and display them individually. This is an example from the OptoView RT view. Well, if we look into the next generation of OCT technologies, I think one of the most promising areas is a new type of Fourier domain detection, so-called swept source Fourier domain. This actually enables even faster speeds than the current spectral domain detection, 10 to 20 times faster. It does not require a spectrometer or a line scan camera, and it can image at long wavelengths where we have improved choroidal penetration and immunity to ocular opacities. It does, however, require laser technology, narrow line with high-speed frequency swept lasers. So again, this is borrowing from advances in the photonics and telecom communities. So with a swept laser, we can achieve extremely high imaging speeds, which would enable wide field retinal imaging. This is an OCT fundus image from a volumetric set. It's in fact acquired at 400,000 axial scans per second. That's more than 10 times faster than most clinical systems. In this case, this is motion corrected using software, and just to show that it's a volumetric image, we can extract multiple cross sections at an arbitrary position in this volumetric data set. So this promises to both simplify the clinical examination protocols, but also provide comprehensive information on retinal structure. I wanted to mention also that you can notice extremely good choroidal penetration with the long wavelength imaging. Here's another example focusing on a smaller region, the optic nerve head. In this case, we're scanning an en face plane progressively deeper into the nerve head. This shows structure of the optic disc as well as the lamina crebosa here. And this is a potentially powerful approach for early diagnostics and assessment of glaucoma progression. Another advantage of swept source imaging is that we can have extremely long imaging ranges. So this shows an example of anterior segment imaging where one can image the corneal lens in the anterior chamber. This is at 200 kilohertz axial scan rate. That's, again, faster than commercial systems with quite a long imaging range spanning the anterior cornea to the posterior lens. By using this type of information, one can obtain comprehensive optical biometry on the anterior chamber, that is, measurements of topography, corneal thickness, corneal power. And these types of measurements could be quite important for assessing intraocular lens power, especially in patients who have had keratorefractive surgery. We can extend imaging ranges to extremely deep depths. In fact, it's possible to image the entire eye length here. This is imaged at 50,000 axial scans per second, going from the front of the cornea all the way back to the retina. This is a rendering. And then here you see an example of a cross-sectional fly-through. These types of images can integrate measurement of axial eye length with anterior chamber measurements. Again, important for keratorefractive surgical planning or intraocular lens implant power determination. With very high speeds, we also have the potential to extend structural imaging to functional imaging. Here we see an example of a volumetric Doppler data set. This also requires very high speed. The advantage of a volumetric set is that we can acquire the Doppler measurement in an en face plane, essentially a plane that intersects the central retinal vasculature. This has the advantage that we don't need the Doppler angle. We can perform a simple and robust measurement of total retinal blood flow. So using this approach, one can repeatedly scan an area spanning the central retinal vasculature. This is scanned at multiple volumes per second. And then by selecting an en face plane, we can measure the Doppler signal in this en face plane, use the pulsatility in the blood flow, and then we can essentially integrate to measure the total retinal blood flow. So here we see an example, the pulsatile total retinal blood flow measured at different points in the cardiac cycle. It's also possible to perform angiography imaging. There are multiple approaches to do this. Essentially, these are somewhat analogous to MR in the sense that one is sequencing the axial scans to measure changes in the image, that is blood flow or other types of microscopic motion. So by scanning different fields, we can have wide field all the way down to a narrow field with capillary level resolution. Here we see an example of multiple volumes that are registered using software motion correction. We're scanning en face down at different depths in the retinal structure. So the advantage here is that we can visualize vasculature in depth, unlike fluorescein, but the limitation is that we don't see leakage. So this gives a sense for, I think, some of the advances in OCT, but I'd like to shift gears a little bit for the last part of this presentation and discuss the translation of technology into clinical practice. I think OCT is an example of many technologies that are poised for translation. So one metric to look at is publications. We can see the growth of publications as a function of time here. You could argue that publications are a surrogate marker for scientific advances in clinical understanding. And publications track, in fact, the availability of instrumentation. So if we look at ophthalmic product development, and in particular the development of multiple organizations which introduced products into the ophthalmic marketplace, we see a corresponding growth in research and clinical knowledge. So I think that this highlights the important role that industry plays in innovation and clinical advances. Similarly, we can look at the role of research funding. The above graph shows cumulative NIH funding in OCT. This is actually plotting programs which either develop or utilize OCT. And government funding is especially important, actually government as well as foundation funding, because it supports a wide range of activities: technology development, translational studies, clinical studies. And clinical studies are especially critical because this provides the evidence on which one can change the clinical standard of care. So we see an increase in funding for OCT and corresponding increases in the number of OCT services. This is actually calculated from Medicare reimbursement. If we extrapolate this internationally, there's an estimated greater than 20 million OCT procedures that are performed every year on a worldwide basis. This is, by the way, from an analysis performed by Eric Swanson, and this is published in OCT News. So if we look at a number of indicators, there's a correspondence between multiple factors here: publications, industrial development, government support, clinical studies. And Mr. Swanson makes the point that the translation of research to the clinical practice really requires this ecosystem of multiple advances: government research, fundamental research, government funding, industry, and clinical studies. So we can go back to OCT publications, and this actually shows the volume of OCT publications stratified according to area. The green bar here is ophthalmology, which is the largest application for OCT. And if we look actually a little bit further down, we see cardiovascular imaging, which is an emerging application. There are about 100,000 cases that have been imaged using intravascular OCT to date. And I think this is poised for significant advances. If we look further down, though, we see a number of other areas which potentially can be developed. And I think it'll be interesting to see how these areas advance in the future. So summarizing then, well, OCT is one example of many technologies that are poised to have widespread research and clinical impact. And I think one of the important messages is that translational biomedical research really requires a collaborative, multidisciplinary effort by people in different communities. And the translation from research to clinical practice really requires a complex ecosystem where we have simultaneous advances in fundamental research, industrial development, government support, clinical validation, and I think entrepreneurship also plays an important role in this. And I guess one point to comment on now, we're in general under pretty significant competitive pressure, both from government funding adjustments, which are common now, unfortunately, in Europe as well as the U.S. Competition can certainly improve quality and effectiveness, and it can also drive innovation. We see similar kinds of cost pressures in healthcare delivery and industrial development. But I think this also causes a lot of stress. And one of the important points to keep in mind, I think, is that we're all working for the same objective, really, to improve healthcare, which has really the benefit that we can reduce human suffering and improve the quality of life internationally. So I'd finally like to acknowledge all of our collaborators. Again, we've been very fortunate to have an excellent collaborative team spanning multiple institutions as well as industry. And then finally, to acknowledge our funding sources, especially the National Institutes of Health and the Air Force Office of Scientific Research. We appreciate the loyalty of our sponsors throughout this entire process. So thank you very much for your attention.
President of the ARVO, Ben Ko, Chairman of the ARVO Foundation, Nicolas Bazin, Chair of the Antonio Chamapalimaud Vision Award Jury, Alfred Sommer, laureates, ladies and gentlemen. It is a great pleasure for me to be back for this year's ARVO Chamapalimaud lecture. I am delighted to once again be able to reflect with you on the remarkable achievements of a group of scientists who were distinguished with the 2012 award and whose work we are celebrating today. As I am sure most of you know by now, the Antonio Chamapalimaud Vision Award was created with the aim of stimulating the fight against blindness. Our goal was and is to single out human vision as a privileged area of action and research. So, our relationship with ARVO, where the award was first launched in the US in 2006, appears naturally and highlights our common interests. Since 2007, we have bestowed 6 €1 million awards on organizations and labs identified for their outstanding contributions. In partnering with ARVO, we bring the true exponents of excellence in your field to one of its largest and most important fora. It is therefore with considerable pride that I am able to be here once again to celebrate the ongoing excellence of our Vision Award laureates. The award has been seeking to recognize the widest spectrum of efforts taking place in the fight against blindness. Our jury panel has reviewed candidates whose work ranges from the most sophisticated research to the farthest reaching use of science in disadvantaged areas of the world. As a research foundation, we might have limited ourselves to recognize only the best research. However, blindness in the world today is so unevenly and unfairly distributed. We decided to treat initiatives to fight blindness and visual impairment in disadvantaged areas on equal foot with scientific discovery. Blindness, preventable blindness, is in fact still today a very serious public health issue in many parts of the world, where it contributes heavily to perpetuate poverty and human suffering. Therefore, our award has a dual focus. In even-numbered years, research is to be considered. In odd-numbered years, it recognizes organizations fighting blindness in developing regions of the world. As our award was conceived in 2005 and 2006, we aimed to contribute to the Vision 2020 goals of the World Health Organization and the International Agency for the Prevention of Blindness. We are now nearing the halfway point on our journey. After 6 full editions, we are able to reflect on the range of scientific and humanitarian endeavors which have already been recognized and received our support. Let me remind you of the identity and diversity of the awardees. Some of them were already mentioned by Alfred Sommer. The Aravind Eye Care System, an Indian organization fighting blindness in Asia and beyond, in 2007, Helen Keller International, a global organization working in many parts of the world, in 2009. The African Programme for Onchocerciasis Control, a coalition of governments, international organizations and private initiatives, in 2011. These were all recognized for extraordinary efforts in delivering healthcare solutions in the developing world. Now, the research awards, whose winners were named by Alfred Sommer, so Jeremy Nathans and Kindwa Yao in 2008, Anthony Movshan and William Newsom in 2010, were recognized for their outstanding research, contributing, as has been stressed, to our knowledge and understanding of vision, from processes in the retina for the first two, to visual processing and perception that occurs in the brain for the latter, all this in 2010. Now, what we are celebrating today, so regarding 2012, in the 6th edition of the award, which we are celebrating now, our jury chose for the first time outstanding research which has proven clinical applications. This diversity of projects in the first half of the life cycle of the António Chamapalimaud Vision Award is a true reflection of the philosophy and ideals behind this prize. We owe a debt of gratitude to our truly exceptional jury panel, led by Alfred Sommer, who each year work very hard to identify the most deserving recipients. We also have the pleasure of having some other members of the jury here, as was already stressed, and I want to thank you all, and to thank also the members of the jury who could not be here today with us. So in 2012, the award recognized two sets of achievements of huge importance to science and medicine. Optical coherence tomography and adaptive optics have both transformed the way we are able to look at the eye. The collaborative efforts of Drs. Fujimoto, Wang, Pliafito, Schumann, and Swenson have generated the development of OCT as a tool capable of imaging the retina with remarkable resolution and is now considered a standard part of care in ophthalmology. Likewise, the work of David Williams in applying adaptive optics to the eye has brought retinal cells into sharp focus and it is offering novel insights into retinal changes caused by aging and disease. Although two very distinct pieces of work, the research carried out by the 2012 laureates shares a number of common traits. Both are, of course, outstanding examples of the power of collaborative effort in science. Both have relied on multidisciplinary approaches which not only have great intrinsic scientific interest, but have been able to offer physicians tools which can bring great benefits to their patients. It's also true that we have yet to see the extent of the influence and reach of these tools in medical science, separately or used together. The work of David Williams, James Fujimoto, David Wang, Carmen Pliafito, Joel Schumann, and Eric Swenson will live long in our memory, and the impact of their achievements will continue to grow over the coming years. We are now well into the year 2013, and it will soon be time to celebrate the worth of the forthcoming laureate. This year, the award will again celebrate the fight against blindness in the developing world. The recipient will be announced in the late summer, and our jury is choosing from a truly large and exceptional pool of candidates. Let me now, as Alfred already did, take advantage of this occasion to remind you that we are now requesting nominations for the 2014 award, which will, as in 2012, recognize scientific achievement. All currently active and productive groups in vision research whose work deserves recognition can be nominated. The €1 million award is designed to further the resources of their research and can be used in any way that advances their goals. All of you in this room surely know worthy candidates, and we rely on you, scientific community, to bring them to us. We welcome entries from all over the globe. As someone from a non-scientific background, I am constantly inspired by the vision and commitment of individual researchers. We cannot overstate the role of science in fighting poverty and disease, and in improving the lives of millions of people who will never get to know the names of scientists whose work they are benefiting from. Without them, our world would be a very different place. Without you and your colleagues, no audacious goals, Dr. Paul Siving, could be reached. I hope the Antonio Chamapalimaud Vision Award fulfills its role in promoting the advancement of science and in recognizing the irreplaceable role of scientists. Thank you.
Let me thank all of you for joining us this evening, and I look forward, obviously, to seeing many of you tomorrow, but having the celebration of the Chamapalimaud Vision Award next year at this time. Good evening.